Oh, hey Bio30s. I thought I'd slow things down a little bit as we talk about gene expression. So two key topics to go over today, transcription and translation. So we're going to talk about how do we take our DNA and build proteins from that DNA. So the best way I like to think of it is, say you're building IKEA furniture, right? You have your instruction manual, you have your parts, you use the instructions to put the parts together to build your piece of furniture, right? You don't actually use the physical instructions to build that furniture. So your DNA, you can think of as the instructions. You don't actually take your DNA and mix it together to make a protein. It's just the instruction, it's just the code. The bits and pieces to make a protein, they're called amino acids. So basically the DNA is telling you what amino acids to put together to build certain proteins, and that's what we're going to discuss today. So when we talk about why proteins are important, it is a few reasons. Number one, they make up structural components of a lot of cells. Cell membranes, organelles, these are all proteins, very, very important. Proteins make up our muscle fibers that allow us to move. When we think of proteins, this is probably number one here. We think of building muscle and things like that. Appearance, your hair color, your nails, these are all proteins. Enzymes that speed up reactions in our body, these are proteins. Antibodies that help fight off infections, these are proteins. And finally, hormones that we talked a lot about in chapter 15, they too are proteins. So all of these proteins are made using amino acids, using DNA as the blueprint. So this is kind of the central dogma when it comes to how we move information. So how do we go from DNA to proteins? Well, DNA, as we know, will be able to replicate itself over and over again within a cell during the S phase of interphase. That's just a straight replication. Nothing is actually being produced in terms of proteins or things like that. You're just making copies. What we want to talk about today is how we go from DNA to RNA, which is the process of transcription, and then how we go from RNA to a protein, which is the process of translation. So let's start off the importance of DNA and protein synthesis. So it is not the sugar, it's not the phosphate, it is the nitrogen bases that determine which protein will be made. So it's the guanine, it's the cytosine, it's the adenine, and it's the thymine. It's those four nitrogenous bases that make up our DNA molecules. This is also very important to note. DNA does not leave the nucleus. It's a very large molecule, and it's far too big to leave the nucleus, and honestly, we probably don't want DNA leaving the nucleus anyway, right? You want to keep your instructions in a safe place. So instead, we must use a complementary code or a different way of getting the instructions out of the nucleus, and that's where the mRNA comes in, the messenger RNA. Now, the difference between RNA and DNA, they're very similar, but there are some key differences. Number one, RNA, the R stands for a ribose sugar, as opposed to a deoxyribose sugar. RNA actually doesn't have any T's. There's no thymine. Instead, the thymine is replaced by a nitrogen base called uracil. And finally, RNA is single-stranded, meaning it's just one band or one strand of RNA. There's no two sides to it. How DNA is double-stranded, that double helix. RNA is just a single strand of molecule. So, three types of RNA that we're going to discuss today. mRNA, which stands for messenger RNA. rRNA, which stands for ribosomal RNA, so RNA within a ribosome. And tRNA, which stands for transfer RNA. So keep those three in mind as we go through this video. They're going to be very, very important as we go along from DNA to a protein. So I think we're ready to start our first process, which is called transcription. So transcription, the best way I can describe transcription is this, okay? So let's say, for example, you're starting up a cell phone company. So you're starting up a cell phone company and you're trying to compete with, say, the big players here, Samsung, Apple, Google, etc. So what you're going to do is, instead of inventing the best brand new cell phone you can, you can't come up with any ideas, right? All the great ideas have already been taken. So instead, you're going to go see what Apple has to offer. So you're going to break into Apple's headquarters, you're going to see the plans for the new iPhone, and you're going to jot down those plans in your notebook, because you can't steal the plans. If you steal the plans, they know you took them. But if you jot them down in your own words and know how to build them, then they wouldn't be any wiser. So this is what transcription is. I don't know if it's the best example or not, but transcription is looking at DNA and doing a shorthand version of that, so writing down a shorthand version of DNA in the form of mRNA. So we need to take DNA, make it small enough to leave the nucleus in the form of messenger RNA. So what we're going to do is we're going to have an enzyme come in, RNA polymerase. And the RNA polymerase, this big enzyme here, is going to lock onto a promoter region. This is step one. It's called initiation. So it locks onto a promoter sequence. This is not transcribed, it's just where the enzyme hooks onto, okay? Think similar to the primers when we talked about DNA replication. So enzyme locks on to a promoter sequence, this is initiation, and then as the enzyme reads the DNA, it's going to before it reads the DNA, it's going to unzip the DNA, much like helicase does. So it's going to unzip the DNA, and then it's going to start building a chain of RNA. So it's going to match up the pieces here with the appropriate RNA. So C's go with G's, A's go with T's, but there's no T's in RNA, so it would be a U that would go with an A. So anytime you would see a T, you would replace that with a U, a uracil. So then we have CAU, CCA, and away we go. As we build this chain of mRNA, this is called elongation. So the RNA polymerase binds to the promoter region. It starts building this chain of mRNA. This is called elongation until it reaches step 3, which is termination. And once we reach a termination point, the enzyme knows to detach from the DNA. The DNA reforms those hydrogen bonds, so nothing is destroyed or broken. And now we have a complete strand of mRNA. Transcription is now complete. You've now completed your notes. Now, in reality, it's not that simple. There's other stages involved where we're actually splicing and cutting out bits and pieces of DNA, adding things, preparing it to leave the nucleus. But you guys don't have to know that for this course. So, ultimately, RNA polymerase comes in, transcribes a strand of mRNA, elongates it until it reaches a termination point where we now have a full strand of mRNA, okay? A reminder that when you do this, say in a practice question or an exam, you would not be placing thymine in there because there's no T's in mRNA, there's only uracils, so there's still G's, C's, A's, and now U's. So that's essentially half of protein synthesis. So we've done half of protein synthesis. We've now gone from DNA to mRNA. Now we need to translate the mRNA into a protein. So we need to take those stolen cell phone plans that we wrote down and now use them to build a cell phone for our brand new company. And that is protein synthesis. Now, we're here, right? We went from a DNA strand to an mRNA strand. How do we go to a protein strand? So how do we go from this to this? How can you code for 20 amino acids in our body with only 4 letters? So how do we use A, U, G, and C to make over 20 amino acids? And how do we know that A, U, G gets us the amino acid methionine? And how do we know that G, C, G gets us the amino acid arginine? Well, there's a few ways that we do this. Number one is the mRNA is read in triplets or groups of 3. We call these codons. So the triplets are codon 1, 2, 3, 4. So we're reading them in groups of 3. So UGG, UUU, GGC, and away we go. And one codon equals 1 amino acid. So essentially it's like a 3-letter word leading to a amino acid. So we read them in groups of 3. So we read them in groups of 3 to get to our amino acids. So for example, AUG gets us MET, CGU gets us ARG, and away we go. Now, do you have to memorize all 20 amino acids? The answer is no, you don't have to memorize all 20 amino acids. Do you have to memorize what makes all 20 different amino acids? The answer is no. Luckily for you, and this is included in Microsoft Teams and Moodle, you have this essentially the code for all life, right? The code breaker, if you will. So with this chart, you can use this codon chart to figure out what any codon out there, what it will code for. Because the code is redundant, meaning more than one codon will code for multiple amino acids. So for example, let's just pick one here, leucine. 1, 2, 3, 4, 5, 6 different ways you can make leucine. Why is that important? Well, because if there's any issues or mutations or mistakes made, let's say for example you were supposed to have CCU, but you ended up with CUC, does the amino acid change? No, it doesn't, and then the protein will function just fine. So if there are mistakes made, it's okay, sometimes. Also, this applies, right, because remember that DNA is found in all living things, so yes, the code for DNA is universal, but the sequencing and the genes present are different between species. Couple things to note here, a start codon is AUG, meaning your sequence will always start on AUG, and a stop codon is either UGA, UAA, or UAG, meaning once we reach a stop codon, the process of translation is over. So let me just quickly show you how to use this chart in case you have to go through questions with it. So let's pick a random codon here, it could be anyone. Let's do GCA. That's our codon, GCA. So here's what I'm going to do. I'm going to look on the side here that says first base pair, and my first base pair is a G. So I'm going to go down here and find G. My second base pair, which is up here, is going to be a C. So I'm going to go up there and circle C. And my third base pair is going to be an A. So now I simply go G, C, A, meet up down here, and then here's the amino acid that it codes for, alanine. So that's all you have to do. Quite simple to use this chart, although if you're coding proteins that are a few amino acids long, it might take you a little while to make sure that you get all the amino acids correct. But as long as you just follow first, second, and third, you should be able to get all the amino acids within that protein. So, how does the process actually work? Well, here's what happens. We're going to have that mRNA strand that has left our nucleus, and it's going to come in contact with a ribosome. And a ribosome will have two parts, a large subunit and a small subunit. And within the ribosome, we have our rRNA, our ribosomal RNA. And the ribosome will literally clip onto the strand of mRNA. It will attach to that strand of mRNA, clip on there, and begin coding from AUG. So it's going to begin coding from our start codon. As the ribosome moves down the mRNA, it's going to read each sequence in codons. So it's going to read each section in groups of 3. So it's going to read our start codon, then codon number 2, then 3, then 4, then 5. When it reads a codon, it sends a message to pick up the appropriate tRNA, which is the transfer RNA. And the transfer RNA will bring in the appropriate amino acid. So say, for example, we wanted AUG, which is our start codon. So I'll use AUG as our example, our start codon. Oops. So here's AUG, okay? The anticodon is the reverse of the codon. So the tRNA contains what's called the anticodon, and the anticodon will be the matching pair to the codon. So the anticodon to AUG would be UAC. So that's our anticodon, forms a little hydrogen bond there. And then we have our tRNA. I'm not going to try and draw the tRNA here. And at the very end of the tRNA, we have our amino acid. So now the codon and the anticodon bind together, bringing in the amino acid. Then when that's done, the amino acid stays because the next tRNA will come in. So the next tRNA will come in, bringing in the next amino acid. And then the third tRNA would come in. And then as a new tRNA comes in, the old tRNA leaves. And it's just a shuffle. So we shuffle down the row. As the ribosome reads the mRNA, we shuffle down and make an elongated chain of amino acids. So you'll notice that these processes are similar, right? They're pretty much identical in terms of what's happening. In transcription, we had an initiation, an elongation, and a termination. In translation, we have an initiation, an elongation, and then eventually we have a termination. So we're going to keep elongating this strand of amino acids, building our protein, one at a time. You guys see how the tRNA, number 2, number 3, and then number 4, we're continually building our chain as we bring in new tRNA, as the ribosome moves down, just kind of like reading it like you would say ticker tape. And we're going to place a new amino acid at the end of the structure. And we're going to continue this process of elongation until we reach one of those 3 stop codons. And when we reach a stop codon, we stop the process. So now the ribosome detaches from the mRNA, they go about their way, and then we're left with a newly synthesized protein. So now we have made a brand new protein. We have finished our IKEA furniture, or we've made our brand new cell phone, and away we go. So putting it all together, initiation, we started a start codon, we bring in that starting amino acid, methionine. Elongation, we continue to bring in amino acids one at a time, one at a time, one at a time, building this chain, binding them together with peptide bonds, forming a polypeptide, and finally we reach the termination stage where the ribosome detaches and we have a fully formed polypeptide of protein. All right, that's a lot to take in, so if you guys do have to rewind this video or replay some parts, please do. But thanks again for watching, and next episode we're going to talk about some biotechnology, so how we can actually change and mess around with DNA in various ways, and number 2, how DNA can actually change in the form of mutations. So, thanks again, and we'll chat soon.